The cell cycle, presented by Group 10. Scientists initially saw two phases of cellular life under the microscope. These were termed interphase and mitosis. Interphase was later broken down into the growth 1, synthesis, and growth 2 phases. These are major checkpoints during and after mitosis. These factors are present to ensure the cycle is running smoothly. Tyrosine kinase receptors bind to the growth factors to signal cell division if everything is normal. Cyclin-dependent kinase is the key protein in triggering different steps in the cell cycle. In 2001, Leland H. Hartwell, R. Timothy Hunt, and Paul M. Nurse won the Nobel Prize for this discovery. The kinase is inactive unless there is a cyclin that attaches and phosphorylates the inhibitory phosphate. There are different cyclin CDK complexes that trigger different steps in the cell cycle. The cyclin concentration cycle is another step in determining what part of the cycle the cell is in. During interphase, cyclin concentration is very low due to synthesization. During mitosis, concentrations greatly increase, and after mitosis, the cyclin is degraded. The cell synthesizes mRNA and proteins. The cell also grows in size and starts to create new organelles such as the mitochondria and ribosomes. Towards the end of the G1 phase, growth-dependent cyclin-dependent kinase activity increases, which promotes DNA replication and initiates G1 to S phase transition. At the G1 checkpoint or restriction point, the cell either commits to division or exits from the cycle and enters the G0 phase. Then, the cell will only move from G1 to S if it is an appropriate size and has adequate energy reserves. If damaged, P53, a transcription regulator, activates the transcription of gene P21. Transcribed protein P21 prevents cells from going into S phase. This gives time to repair the damaged DNA. P53 can also order the cell for apoptosis, cell death. Following the G1 checkpoint, the cell proceeds through the S phase and reaches the G2 checkpoint. This checkpoint bars cells from entering mitosis. If the DNA is damaged, the cell will not enter mitosis, but will attempt to repair the damaged DNA. After the DNA is repaired and fully synthesized, the cell will be able to enter mitosis. Mitosis is a four-stage process in the cell cycle. It occurs right after the DNA replication that occurs in the G2 phase. The first stage in mitosis is known as prophase. The second stage on mitosis is known as metaphase. The third is known as anaphase, and the final stage in mitosis is known as telophase. Prophase is the first and also the longest stage of mitosis. In prophase, the chromosomes fully condense. The centrioles also move to opposite ends of the cell.
Created using Powtoon.